Hey, it's Moser with a very quick genetics review. We're going to look at Punnett squares, both simple ones and complex ones. So let's start off with a few just simple review facts. All organisms have traits, and those traits can include things like fur color, eye color, hair texture, wing pattern, toe webbing. You name it, any trait that an organism has, well, we can name. And even better, we can find the genes that control it, because that's right, traits are controlled by genes. And you remember that genes are actually just sections of DNA that code for specific proteins. You should also remember that many genes have two versions called alleles, and that alleles generally come in a dominant form and a recessive form. Dominants are shown with a capital letter, recessives are shown with a lowercase. And for any allele, everybody gets two copies, two versions, and they could be the same and they could be different. We don't know, but where'd you get them? Well, you got one from yo mama and you got one from yo papa, hence two. Now, you may also remember learning that if you've got at least one version of one of the dominant alleles, you'll have the dominant trait, and if you have both copies of the recessive allele, you'll have the recessive trait. We generally, in middle school, grossly oversimplify this and teach things like brown eyes and blue eyes. Really, eye color is way more complicated, but that's okay. We'll go with the simplified version you learned before for now. Now, you may also remember something that looked like this. People call it, what's that, the gene box, the allele thing? Yeah, it's a Punnett square. And when you did Punnett squares, you distributed the alleles based on what a parent could possibly contribute. For instance, if we've got a mother who has a big B, little b with her lovely brown eyes, that is, she's heterozygous for eye color, every time her body makes an egg, she could contribute a big B or a little b to that egg. If we have a dad who is little b, that is recessive homozygous, every time he makes a sperm, it's going to have a little b. We then use the Punnett square to figure out the possible genotypes of their offspring. Remember, genotypes are the actual genes you have. And from that, we can figure out how many individuals or what percentage of individuals of their offspring would tend to have what genotypes and what phenotypes. In this case, with this cross, we would expect that roughly 50% of their offspring would have a big B, little b genotype, and 50% of their offspring would have a little b, meaning 50% of their offspring should have brown eyes and 50% should have blue. Now, that was a gross oversimplification, but the other thing that we've simplified is this fact. Genes, well, you've got more than one set of alleles. You've got more than one trait. Everything is made up of more than one trait. So, we're going to step it up and look at some dihybrid Punnett squares. Hey, let's talk about guinea pigs. Aren't they cute? You know, they eat them in South America, but that's a whole nother story. So we're going to look at two specific traits in guinea pigs: smooth coat and rough coat, and long coat and short coat. Smooth coat, smooth fur, rough fur, short fur, long fur. These are controlled by individual genes. The long fur, short fur gene is usually written with a capital L for long fur because that is dominant, and a lowercase l for short fur, which is recessive. And the rough fur, smooth fur is written with a capital R for rough and a lowercase r for smooth. So let's say that mom is heterozygous for both traits. She's got a big L, little l, big R, little r. And so's dad. Wow, that's weird. So let's figure out what genes, what alleles each of these parents could contribute to the gametes or eggs and sperm that they produce. Well, we know that mom is going to manufacture eggs inside her body, and every time she does, there's a 50% chance that those eggs will receive a dominant L for long fur, and there's a 50% chance they'll receive a recessive small l. For every large L egg, there's a 50% chance that they'll receive a large R and a 50% chance they'll receive a small r. Same for those little L eggs, 50% chance of a large R, 50% chance of a small. That means we have 4 possible combinations for just these two alleles. Well, what about dad? Since dad is also heterozygous for both traits, Again, there's a 50% chance of him receiving a large L or a small L, or the sperm receiving a large or a small L. And for each of those, there's a 50% chance on the R's as well. What does this mean? It means that we've got 16 possible combinations of these two genes for these two parents. So we've lined up a dihybrid Punnett square. We've arranged all the possibilities for mom across the top. No, you don't have to draw eggs when you do this. And all the possibilities for dad down the side. With this information in hand, we can go through and tally and see what percentage of their offspring we would expect to be homozygous dominant for both characteristics, big L, big R, little r, etc. And then we can look at their phenotypes. Remember, phenotype is the physical characteristics that the genes create in an individual. What we find is that for these parents, over half of their offspring should show the dominant phenotype for both traits. And only 6% of their offspring are going to show the recessive characteristic for both of their traits. About 20%, 18 to 20% of their offspring each should be dominant for one and recessive for the other. Now, why use a tool like a dihybrid Punnett square? Well, as fun as it might be to predict that about half of the offspring of a particular parent cross would have long, rough coats, it's not really the reason we do dihybrid Punnett squares. We do them specifically to look at these percentages. When you watch the next video, you'll understand that it's not always a 50-50 chance that any gamete containing one allele gets either of the others. So keep those ratios in mind, keep the percentages in mind, and realize that we don't just do dihybrid Punnett squares for fun.